All right.
Okay, welcome everyone today. Hope you all are having a good Monday. I'm going to do an introduction of the NeuronS Matter Research Network before we hear the talk for today. And who we are is we're an inclusive, diverse community of neuroscientists, engineers, physiologists, manual therapists, and massage clients. And we are a U24 of the National Institutes of Health in the United States, funded by NINDS and NCCIH. And you can see our contact information there on the slide, and I'll come back to that in a second. So our leadership team consists of Dr. Ellen Lumpkin and I as co-PIs. I'm Greg Gerling, along with the other members that you see from around the United States and around the world who are on the screen right now. Our network intends to nucleate an emerging field focused on the neurobiology of soft tissue mechanotherapies by bringing together some of these diverse communities you see right now, where different tools, whether they're experimental, whether they're computational, whether they're clinical, are brought together from neuroscientists, physiologists, engineers, and clinical scientists and manual therapists. And what our high priority goals are is to establish an inclusive research network to identify mechanisms through which soft tissue manipulation, such as massage, exerts biological effects on the nervous system as well as non-neuronal cells and tissues. And we have three main high priority areas. One is mechanistic multi-scale research measuring the physiological response to force-based manipulations in neural and non-neural cells and tissues. Second is a mechanistic understanding of the neurobiological basis of the psychosocial sense of affective touch and how affect mood modulates neural and physiological responses to mechanical stimuli. And finally, in technology and methods development for mechanistic studies, including computational approaches. And so our network has three kind of main thrusts, as well as the diversity, equity, inclusion, and belonging component as well. And these are three of them. The conference program. And so we'll be putting on scientific conferences that are cross-disciplinary in years 1 and 4 of this effort. This is a 5-year effort, hopefully to continue beyond that. We also be hosting summer schools, and the first summer school is going to be announced fairly soon. It's going to be in Indianapolis in the summer in June. We are going to be having funding travel awards for inclusive excellence. In fact, we've already started to do that. And then we are hosting a virtual speaker series, which is why you're here today. And this is the first speaker. There's also a pilot project program where we'll be funding projects. And there's a request for proposals that will be coming out fairly soon on that, as well as mini sabbaticals to allow researchers to move and have residencies to attain technical skills or perform experiments. We've already started to do that as well. Finally, we are establishing various dissemination means, a website, social media, YouTube channel, and open access initiatives and others. And so please look for these new initiatives to start to kick in soon. So we would encourage you, please join us. We're looking to increase our membership. And if you're interested in joining, we'd love to have you. We'd love to have you involved, and just say how you would like to be involved. And I'm sure we'd really love that. The contact information is on our website, massageneuroscience.org. And you can also subscribe to the Twitter accounts as well, NeuronS Matter. Okay, so as I mentioned, this is our first speaker in our virtual speaker series. We're really glad that the first speaker could be Victoria Abraira. She is a co-I on this team. She is in cell biology and neuroscience at Rutgers University. She uses molecular genetics to identify and manipulate circuits of touch. And her topic today is going to be on wired for touch, the neurons and circuits of the somatosensory system. Please welcome Dr. Abraira, and we're looking forward to her talk.
Thank you, Greg. Now I'm going to go ahead and share my screen. Okay, give me a moment. All right, so you should all see my name as my title slide. Okay, great. So thanks, Greg, for the introduction. I'm so happy to be the first. And I know that we have lined up exciting speakers for the rest of these virtual seminar series, or at least we're thinking about it and putting it into action. But I'm really honored to be the first. So our brains have evolved for us to thrive in a very complex world, and we can never really fully appreciate all the complexities of our environment, but we can approximate salient features through the use of our senses. Now, skin is the largest sensory organ of the body. And while it's very thin, it's just a few millimeters thick, it's very durable and it's very flexible. It gives rise to our hair and our nails. It has an immune function and a very important barrier function. And as human species, I think we really love our skin. We glorify it by tattooing it and piercing it. We take care of it. We wash it, we massage it. Some of us even remember to moisturize it every once in a while. But as a neuroscientist, I think that skin is remarkable because it harbors the sense of touch, which is arguably the sense that we know the least about, in part because of the complexity of the peripheral nervous system, the skin regions that it projects to, and the brain regions that then sends this information to. But in spite of its complexity, I think everyone here can agree that a simple act like caressing of skin can at times be different enough for us to deem certain touch unpleasant and some to be quite pleasant. And I would argue that it's in this unique ability in which we qualify touch that bring out some of our most human of characteristics. So how does skin accomplish this? Well, skin is not uniform. We have glabrous skin, so that's the skin on the palms of our hands and feet. But the large majority of skin, over 90% of it, is actually hairy skin. Now, whether you have a lot of hair or none at all, it's all hairy skin. And embedded within skin are mechanosensory end organs like the hair follicle, or for example, the Meissner corpuscle or the Merkel cells that are innervated by specialized sensory neurons that we call the low threshold mechanoreceptors, or LTMRs. And the first step leading to the perception of touch requires this activation of these LTMRs, whose cell bodies reside in the dorsolateral ganglia. We have 32 pairs of these ganglia that line our spinal column. And all LTMRs have a branch that, of course, extends to the skin, and another one that penetrates the spinal cord. Now, a subset of these LTMRs also have a branch that ascend via the dorsal column to the brainstem. And these specific LTMRs are one of the longest neurons of the body. So imagine an LTMR that innervates the tip of your toe, sends a projection all the way to your hindbrain. And for some of you, that could be a neuron that is 6 feet long. So that's quite remarkable. Now, from the brainstem, touch information goes to the thalamus, and from there it ends in the somatosensory cortex. And this is, of course, the anatomical basis of the direct pathway, which has really dominated the textbook view of touch perception, which basically says that there's a label line of modality segregation from the skin all the way to the brain. However, there's a second pathway, which is this indirect pathway, which has these projection neurons that carry process information from the spinal cord to the brainstem. And when I started as a postdoc in David Ginty's lab a few years ago, there were several fundamental questions about the touch system that were unanswered. So in hairy skin, we wanted to know what is the functional organization of all these different LTMRs and what features of the tactile stimulus are they extracting? And within the central nervous system, what is the relative contribution of the direct versus the indirect pathway to touch perception? And when it came to this indirect pathway, the spinal cord, how are LTMR inputs organized? And does this indirect pathway contribute to tactile perception and tactile-based behaviors? And if so, how is LTMR information represented and processed by the spinal cord? And during my postdoc, I addressed some of these questions using the mouse as a model system with these publications. So the first two are related to the organization and function of the LTMRs in hairy skin. And the third one address the contributions of the indirect pathway to touch perception. I like to sort of highlight this review that proposes areas of research that now my lab is starting to study. And lastly, I would also like to point out this collaboration with Bob Dada on the use of machine learning to objectively scale behavior, which becomes important later on in my talk. So let me first really review briefly what we know from hairy skin. So it was really Zoderman and colleagues back in the 1920s, which used amplifiers to really record from these nerve bundles and establish that these LTMRs can be grouped into different categories based on things like conduction velocities and action potential waveforms. So for example, you have the very slow C LTMRs and the very fast A beta LTMRs, as well as their adaptation properties to the sustained mechanical stimulation. So you have some of these LTMRs that are slowly adapting, they respond throughout that touch stimulus, and some that are rapidly adapting, they just respond to the on and off properties of that touch. And when I started as a postdoc, our basic hypothesis was that these neurons that are not only distinct in their physiological properties, but also in their molecular properties, the expression of genes that give rise to these different electrophysiological properties. And we use this hypothesis to build a variety of mouse genetic tools that would allow us to generate pictures like this, finally visualizing physiologically distinct LTMR endings in mouse hairy skin. And to summarize what this large body of work showed, and I'm not citing everybody that's contributed to this picture, but there's been several labs that have contributed to this, to the fundamentals of what I'm about to say. We really are understanding how each type of hair follicle, we have 3 different kinds, act as a sort of mechanosensory end unit, each receiving unique combinations of these LTMRs. And what we showed was that this organization is absolutely crucial for each LTMR to extract unique features of the tactile environment, like, for example, the direction of hair follicle movement, indentation, velocity of stroke, stretch, etc. So in essence, what we collectively found as a field, how each LTMR really acts as an individual instrument in a large orchestra. And each one is tuned to specific features of that tactile stimulus. And each articulates this message to the brain via their unique physiological properties. And it's really this collective ensemble of LTMR activities that give rise to these symphonies that our brain has to distinguish between a caress or a poke or a hug, etc. So how are these different LTMR activities encoded by that first relay station, which is the spinal cord? Now, based on label line models, we would think that perhaps each LTMR would have a dedicated recipient in the spinal cord. However, the problem with testing this simple hypothesis is that in comparison to neurons that process pain and temperature information, we didn't know much about the interneurons that process this touch information. So as a postdoc, I set out to build a series of mouse genetic tools that would allow us to know more about them. And this large body of work is basically summarized here, in which I showed different mouse lines that I built. Now, the pain and temperature laminas in blue, those touch projection neurons that send information to the brain are in red, and sandwiched in between those are these interneurons that receive LTMR information. And collectively, they represent about 80% of the spinal cord. And in this study, we use these tools to do really basic things like understand the proportion of excitatory versus inhibitory neurons in this region of the spinal cord. We were able to use these tools to understand basic anatomical and electrophysiological properties. We also set out to build Cree-Flip intersectional studies. Mouse science basically is the way that would allow us to specifically manipulate the activity of these neurons in the spinal cord only, to really answer questions about whether this region of the spinal cord, whether processing of tactile information in this region of the spinal cord is important for tactile-based behaviors. And it is indeed important for tactile-based behavior. So we use these different mouse lines to inactivate these different kinds of neurons and show that mice develop various deficits in tactile perception, which was an important point to establish at the time. And along with these various tools, we set out to build connectivity patterns. So this is high resolution microscopy and circuit electrophysiology to establish really basic maps of connectivity. And basically what we showed is that each interneuron subtype in the spinal cord receives convergent information from these different LTMR modalities. And lastly, by recording from the projection neurons that send information to the brain, we were able to directly test label line models. So I'll summarize it here. So basically what we learned was that LTMR information is not processed via simple label lines, but rather information from multiple LTMRs are sparsely distributed and processed by these various different types of interneurons, whose outputs are defined by sort of these weighted averages of these distinct LTMR modalities. And layer on top of this, we also found that these interneurons are influenced by direct cortical spinal input. So there's a lot of top-down control within this region of the spinal cord. So, to summarize, in the skin, information is organized in sort of this highly structured manner, right? So you have these finite numbers of LTMRs that innervate a finite number of hair follicles and these very predictive patterns. And of course, this organization is absolutely critical for each LTMR to extract these salient features of the tactile stimulus. But in the spinal cord, this information is distributive. So with each LTMR subtype talking to a wide variety of interneuron classes. Now, these simple anatomical findings suggest that touch information that enters the spinal cord is without any inherent value, and that central representations of touch are likely to differ across developmental, emotional, and disease states. And this is why we feel that touch can feel so different depending on your state. Now, these basic observations pose extremely interesting questions that my lab is answering at this time. In the short term, we want to understand how do internal states influence these spinal cord touch representations? And in the long term, how are associations in supraspinal centers imposed onto these spinal cord circuits? And how do these supraspinal centers compute value to elicit appropriate behavioral outcomes? And what I'll talk to you about today is some of the ongoing projects that basically demonstrate the framework for how we are tackling these long-term feature directions. But to put these feature directions into perspective, let me first sort of tell you where the touch field stands. So, and in doing so, I will use an analogy. So half a century ago, or more now, Hubel and Wiesel and colleagues described basic concepts of visual processing in the cortex, like these direction selective responses in V1. And at the time, it was thought that because these features are encoded in the cortex, that they must arise as the result of sort of these intricate cortical connections. But in the shadows of these findings laid the retina, sort of what was thought to be sort of a passive relay of information. But now we have a much greater appreciation of the retina as an important computational powerhouse for the visual system. For instance, in this elegant example of the direction selective retinal ganglion cells. So in essence, I like to propose that the spinal cord plays an analogous role to the retina, representing an important locus for processing, modulation, and perception of the sense of touch, and not just a passive relay point. So this is where I feel that the touch field sort of stands right now, at least in my corner of the world, in which we just sort of uncovered the retina. And what's exciting is that collectively as a field, we can use sort of reverse engineering tools like, mouse genetic tools that allow us to uniquely study the sensory neurons as well as the interneurons and projection neurons. And in our lab, as well as other colleagues in the field, we use intersectional genetic strategies to genetically isolate these different components and really apply an alchemy of anatomy, electrophysiological, and behavioral approaches. For our behavioral analysis, we use a variety of data streams, high-speed camera, depth vision, muscle recordings, calcium imaging, et cetera. And we apply various computational approaches with sensitivities that are specifically designed to match the granularity of our circuit dissection techniques, but also specifically designed to address the question at hand. So my research team is comprised of a diverse and multidisciplinary group of trainees, so computer scientists, data scientists, biomedical engineers, et cetera. The team, the breadth and depth of the team is best exemplified by this next story, which I think really provides the framework of our long-term vision. And this story is spearheaded by a diverse team of postdocs, computational biologists, and gap year fellows, and is supported by the Pew Charitable Trust and NINDS. So we know that touch can make us feel better, especially when we are in pain. And this is because our experience of touch involves, as I said, many brain structures and is shaped by both sensory, emotional, and social factors. However, what we lack is a roadmap of how these simple acts are functionally integrated all along our somatosensory system. And as I alluded to earlier, our sensory systems are under strong evolutionary pressure to develop ways to extract salient features from the environment. So within this large repertoire of LTMRs, we have the subset called CLTMRs that form a unique group that in humans have been shown to be specifically tuned to caress touch or social touch. And my work, as well as the work of others in the field and the genetic identification of these CLTMRs, show that similar to humans, they innervate hair follicles, forming these beautiful endings around the follicle, and have physiological properties that are very similar to that of human CLTMRs. What I'm not showing here is that there are other types of CLTMR-like fibers that are free nerve endings that have been implicated also in massage, in communicating massage in encoding massage-like tactile properties. So we're believing that these LTMRs are a large class, or at least these CLTMRs might be two or more types of LTMRs. And like these direction selective retinal ganglion cells that encode particular features of the tactile stimulus that we believe are relevant to us as social species. So it's not a surprise that we have recently begun to think about the spinal cord projection systems, as these projection systems that carry process information from the spinal cord to the brain, as a sort of eclectic mix of subtypes whose combined activity can transmit both negative valences associated with noxious stimulus, as well as positive valences that are associated with the pleasurable properties of touch. So the question that we wanted to ask here was how does descending modulation, like the type that might be activated during social touch, how is it functionally integrated onto spinal cord circuits to set the valence of somatosensory information via the activity of these projection systems? And to answer this question, we focused on the hypothalamus, which releases a variety of neuromodulators known to sculpt our sensory experiences. And one important molecule with known actions along this positive to negative valence spectrum is oxytocin. So indeed, oxytocin has been shown, or has been shown that oxytocin positive neurons in the hypothalamus are actually activated by somatosensory stimulation on the positive valence spectrum. So this is, massage or caress-like touch. But we wondered whether these oxytocin positive neurons were also activated by somatosensory stimuli on the negative valence spectrum. And indeed, what we showed is that these oxytocin positive neurons in the hypothalamus are activated by noxious stimulus that results from injury. So these are the neurons that can be activated both by social touch, but also by injury. So we wanted to ask whether these neurons actually project down to the spinal cord, and indeed they do. So if we genetically label these descending projections from the hypothalamus, we see that they overlap with spinal cord interneurons that express the oxytocin receptor. And there's this very, very nice overlap between those two. Now, if we patch from the neurons that express the oxytocin receptor in the spinal cord, we see that they respond to oxytocin by increasing their firing frequency. So basically, they become more excitable in the presence of oxytocin. So painful stimuli, we showed that, painful stimuli as well as caress-like touch or massage can activate these oxytocin positive neurons in the hypothalamus. These neurons project down to the spinal cord, and oxytocin excites the spinal cord interneurons that express the oxytocin receptor. So we wondered whether this pathway could promote pain relief. So if so, we would predict that the activation of the neurons that express the oxytocin receptor would relieve pain. And that's exactly what we see. If we inject a virus carrying a conditional excitable dread into the spinal cord of mice in which they have an oxytocin receptor creep, and we inject these viruses in a somatotopically aligned fashion with the injury. So we induce an injury to the leg. In this case, it's a neuropathic pain injury. And in a somatotopically aligned fashion, we inject these excitable dreads. So CNO activation of the spinal cord interneurons that expresses the oxytocin receptor, we find alleviates these sensory reflexive features of pain. So shown here with mechanical as well as thermal stimulation. So after pain, these mice become sensitive to touch, sensitive to temperature. And we see that if we activate these neurons, we can relieve this type of pain. But CNO activation of these neurons in the spinal cord also alleviate more of the affective motivational aspects of pain. And here we showed this with a condition place preference test in which mice on one side receive an intrathecal injection of lidocaine and on the other side intrathecal saline. Now in control mice that are in pain, there's obviously a preference onto the lidocaine side. However, with CNO stimulation of these interneurons, this preference is abolished, suggesting that activating them alleviates pain in a similar fashion, or at least comparable to how lidocaine alleviates pain. So what we showed here is that oxytocin circuits in the spinal cord alleviate both the sensory reflexive as well as the affective motivational aspect of pain. But what is the spinal cord circuit that might be mediating this pain relief? So if we stain for these receptors using yet another mouse line, we see that the majority of these interneurons are located in lamina 2 outer and inner. And this is actually exactly the lamina that receive input from the CLTMR fibers, these social touch sensory fibers. And indeed, we see these direct CLTMR inputs onto these interneurons by histology, as well as electrophysiology. So this is when we can express char-rhodopsin in the CLTMRs and patch from the neurons that are expressing the oxytocin receptor. So we know that there are functional synapses between these two cell types. But then we also find that there's a variety of different kinds of LTMR inputs onto these neurons. And this is very consistent with what we've shown before, in the sense that spinal cord interneurons within this LTMR recipient zone receive a variety of inputs. So these interneurons receive both CLTMR input, a delta LTMR input as well, which work that is ongoing, nociceptive input as well. So we wonder, how does the system integrate sort of these multimodal sensory information? You have touch, you have nociception with descending oxytocin to alleviate pain. And so this is sort of where we turn to other sensory systems to see how oxytocin does this in other sensory systems. So for example, in the auditory cortex, it's been shown that oxytocin acts to regulate the salience of external sensory cues by increasing the signal to noise ratio of the social sensory cues. So basically, by balancing the magnitude and the timing of inhibition with excitation, oxytocin, for example, is able to synchronize a mother's auditory responses to pup vocalizations. And this was shown by Bianca Jones-Marlin and Rob Fromke's lab. And this is a unique feature to experience mothers that have had oxytocin. So we wondered whether this basic model would also apply for the somatosensory system. And if so, we would predict that in the spinal cord, oxytocin engages both excitatory and inhibitory networks. And that's indeed what we see if we stain neurons that express the oxytocin receptor with excitatory and inhibitory markers. We see that there's a split between excitatory and inhibitory neurons. And by electrophysiology, we see tonic and delay firing properties that again are associated with inhibitory versus excitatory neurons. So we believe that interneurons that express the oxytocin receptor are both excitatory and inhibitory. And we're doing additional tests to answer this more directly too. So our working model is basically that by acting on both inhibitory and excitatory network, oxytocin modulates the relative activity of these molecularly distinct ascending pathways that collectively encode valence along this negative to positive spectrum. And we have several ongoing experiments that are directly testing this model. We just got a grant for this, so we're just starting to do some of these experiments. So we're doing some calcium imaging and some various other slice work to show these connections exist. And we think that this model is particularly important because intrathecal oxytocin is currently being used in clinical trials to alleviate chronic pain. And this model would suggest that coupling intrathecal oxytocin with manual therapies, like for example, massage, will optimize the analgesic properties of intrathecal oxytocin. So this is why we think that this model is important. But we're also applying this basic model to other modulatory systems like serotonin as well as cannabinoids, which we feel that these basic models of encoding sensory information along the negative to positive valence spectrum is likely a feature that is shared across all kinds of descending modulatory systems. So this is sort of our ongoing work as other people in the lab are looking into. So obviously these types of models have implications for novel therapeutic approaches to pain management, like for example, these combination therapies. But understanding how activity in these types of circuits in the spinal cord or shape perception requires an ability to scale behavior along these valence states. And in collaboration with Ismail Abdus-Sabor at Columbia and UNC St. John Smith in Cambridge, we're in the process of building a more robust and generalizable platform to assay somatosensory behavior in mice. So current assays like Von Frey and Grimace are very binary, and the only really way to test hairy skin sensitivity, which is what we're interested in the lab a lot, is with the sticky tape assay, where you put a sticky tape on the mouse and see how long it takes to notice it. But with these kinds of outcome measures, you can imagine how translatability is really is really low, you have this narrow dynamic range of these assays, which often obscures drug-specific behavioral effects, which in turn prevents analgesics belonging to different pharmacological classes from being distinguished, or even sometimes our ability to distinguish between an analgesic from an anesthetic. So, so we feel that, I mean, many of us here feel that this is hindrance in terms of the translatability of novel analgesics. But we know that an injured knee can change the way we walk, abdominal pain can change the way we stand, and chronic pain can change the way we interact with our environment and with others. And these are simple acts in nature, but these behaviors can serve as a general guidepost to objectively scale pain and assess analgesic efficacy. So in this study, we used electrophysiology, the electrophysiology of sensory neurons that innervate the injured paw or that innervate the injured knee. So we used two models, the carotid model and an OA model for arthritic pain, to define the time points that punctuate pain progression. And we find that there's increased neuronal excitability that occurs at 48 hours post-injury. However, this excitability sort of resolves itself at 24 hours, matching the increased expression of, for example, chip channels that happens at 24 hours. Now, these types of results, which I'm showing here for carotid, and I didn't have time to put the data that we now have for the OA model, these results are important for two reasons. First, they indicate that adaptations of nociceptive circuits most likely drive prolonged pain behaviors beyond sort of these immediate acute time periods, which we've known before and we've shown for these types of models. But second, I think more practically, it gave us an important timestamp for probing specific behaviors that could differentiate these early to 4-hour from this late 24-hour time point. Now, if we probe sensory reflexive behaviors, sometimes it's really hard to statistically differentiate these two time points. They can be very overlapped. However, if you let the animal sort of run freely and you can start to score ethological behaviors like the amount of time rearing, you start to get sort of that necessary dynamic range to begin to differentiate between these, time points of pain progression. So can we infer pain states by simply observing what a mouse does in an open arena? Now, this is hard for the human eye to do, mostly because mice move very fast. And as predator species, they have evolved to hide pain. So we traded sort of our clipboards and stopwatches for some computers and depth cameras. Now, with depth camera, behavior is collected by the computer as a set of pixels with an X, a Y, and a Z position. So all of a sudden, the problem of mouse behavior becomes more of a computational problem. So much like RNA sequencing, you can use advances in computer inference to reduce the dimensionality of this data. And you can map these pixels into these subsecond epochs. And we could use machine learning to characterize these micro movements or these modules that are millisecond movements that would be very hard for the human eye to see, that are connected in space based on specific transition probabilities. And this is where the machine learning comes into play. So for example, you might characterize a module like this rear. And what you see on the left is a spinogram, which basically is like a line drawn across the mouse to kind of depict, this is how we kind of get a sense of what the mouse is actually doing. And then we can draw a mouse picture accordingly. So you can have something like a rear, module 1, a dive, module 2, and a low rear locomotion, module 3. And again, these are connected in space by specific transition probabilities. In this case, you're more likely to go from a rear to a dive than to go from a rear to a locomotion. And that makes sense because you have to go through dive space in order to get to that low rear locomotion. That's where the arrows in this diagram are showing here. 1 and 2 are more strongly connected than say 1 and 3. Now, this is just what it looks like for these 3 behaviors, but in reality, a 20-minute mouse video looks something like this, in which each circle represents a module connected in space via a specified transition probability. And this is work that was spearheaded by Bob Dada's lab, and I was lucky enough to be around that time to be involved in the process, and gave them a few mouse lines to test their platform. Since then, in my own lab, we further developed this platform to fit the unique questions that we're interested in. Now, based on our previous result, in which we visually scored a change in the amount of time rearing, we first look at the differences in these modules. In a simpler way, are there changes in the usage of these identified micromovements when an animal is in pain? So much like RNA sequencing, you can look at this data this way, in which each module is represented here on a blue, or the module usage is represented here on a blue to yellow scale. Yellow is more, blue is less. And then you have the module number on the right-hand side, and they're organized in this fashion, sort of in these ethological dendrograms, representing families of closely related movements. And so what we see, if you take a bird's eye view with this, is that pain induces profound changes in the usage of these modules at 4 and 24 hours. Now we can have a much more dynamic picture of how usage of the individual modules change from baseline to, for example, 48 hours, in which you have some modules that are used less, some modules that are used more, and some modules that have more of a dynamic picture. But collectively, from a bird's eye view, we propose that is the dynamic changes of these modules over time, which can be used as sensitive behavioral biomarkers for spontaneous pain. Next, we wondered how the usage of these modules might change with an analgesic. So in this paper, we used meloxicam as well as gabapentin for our OA model. And I'll just sort of show you the meloxicams. I didn't have time to update the slides. But by mapping these unique behavioral markers onto behavior space, we see a nice transition from a pre-injury space to a 4-hour to a 24-hour post-injury. Now, giving an analgesic does not mean a return to baseline conditions, but instead it produces an analgesic state that is much closer in behavior space to a pre-injury condition exposed to that specific analgesic. And of course, this has some intriguing implications for our current definition of analgesia. So we show this with meloxicam, but also to a much more severe extent with gabapentin. And this might not come as a surprise to any of you who have been on various pen medications, that opioids can change your general state. So what we show here in mice is that there's an analgesic state in which basically exists in behavior space, and that we have an injury plus an analgesic, it brings you closer to that analgesic alone state. So I began by saying that pain can change the way we behave. And while 3D post-analysis is really good at sort of detecting these subtle changes, like for example, in this diagram that we show here, it cannot itself describe a pain behavior. And that's because pixels are not behavior, right? So behavior, you can think of it as a very complex, and intuitively, we might view it as a combination of these modules and transition probabilities, each individual alone not capable of defining a pain state. So indeed, that's the case. If we train a classifier either on module usage or these transitions, it is at best 80% accurate at predicting these experimental groups. So we wonder whether there was some sort of combination of modules and transitions that might better define these different states. So if you can imagine mouse behavior as a foreign language, what we've been able to do so far is define the nouns and the verbs. But if you think of pain versus analgesic states as different paragraphs, each with their own meaning, we need to start to define, or at least find a way to put these nouns and verbs in the right order to make sentences, and to put these sentences in the right order to make paragraphs. So to analyze our data, we applied an unsupervised sequence embedding technique that is widely used in natural language processing. And when we do this, we find that it's much more accurate at predicting these different pain and analgesic states, much more than the usage and transition alone. But now these embeddings provide us with the means to identify behavioral sequences that characterize different pain states. So for example, we now have these 2, 3, and 4 long module sequences or sentences, which characterize each of the 5 experimental conditions. And when we ablate these very few specific behavioral sequences representing only 0.7% of total syllables, we see a drop in the classifier accuracy of about 7%. In contrast, when the same number of random syllables are ablated, we only see an accuracy drop of 1.6%. So we take these results as a basic proof of concept, not only that complex behaviors beyond these usage and transitions can characterize these different pain and analgesic states, but also that these complex behaviors can be described by using sequence representation methods from machine learning and natural language processing. So now I hope that you can imagine how our conceptual framework and how the spinal cord scales somatosensory information along this negative to positive valence spectrums with our various behavioral approaches represent the basis of how we plan to make headway into these long-term objectives that I spelled out earlier. Now, my talk wouldn't be complete without briefly mentioning other ongoing projects that are related to touch in the lab that really span the skin all the way to the brain. So I'll mention them briefly here for those that are interested in sort of the breadth and depth of our work. So, and I introduced this slide about the direct and the indirect pathway, and there's a lot of questions there that we've yet to understand. So in particular, the dorsal column nuclei. So here, Aman, Mark, and Tom are implementing very simple predictive models to understand how the direct versus indirect pathway, as well as descending inputs from the somatosensory cortex, scale our tactile perceptions from health and disease, in particular in scaling mechanical allodynia after injury. In the spinal cord, we're also interested in very fundamental questions of how touch shapes our movement. And here, Nofar and Mark are bringing forth some really innovative concepts on how tactile information from glabrous skin and joints interface with proprioceptive information from our muscles to set sort of this flexible context-dependent modulation of motor function. This paper is on bioRxiv. And folks like Jack and Elise are really interested in translating some of these basic concepts into recovery from spinal cord injury. And of course, we continue to push the boundary of our understanding of the sensory neurons that innervate skin. And we're partnering with Unilever as well as other foundations to translate some of our findings in mouse hairy skin, including skin from the scalp. And we are trying to recruit people to join our computational team to do some cross-species comparison by histology and single-cell RNA sequencing to try and understand, how conserved are some of these innervation patterns in humans and how it's all put together. And lastly, because Greg mentioned it in the beginning, and then I'll mention it at the end again, in case people kind of jumped on a little bit late. Of course, this is the seminar series, and what we're putting forth today is really a collective of folks, like-minded folks on the Z24 network grant, which we hope will further understanding of how manual therapies, like the type of therapies that most optimally activate these LTMRs that we're interested in studying in the lab, how this results in overall brain health, well-being, and relief from chronic pain. There I have the QR code that you guys can scan that'll take you straight to the website to find out more. And as Greg mentioned, we're doing all of these activities, fun activities, travel awards, etc. So many exciting things that are happening, but I'll leave you with one final statistic, because I'm a sensory neurobiologist by training. For every 100 papers on vision, there's only one on touch. There's an incredible amount of room to expand in this field. And my hope is that people out there start working on touch more to try to understand how it works, how it connects to our brain centers of emotion, etc. So if you're interested in any of the work that we're doing, come join our very dynamic group of postdocs, graduate students, computer scientists, data scientists, and undergraduate interns. It's really their hard work and dedication that make all of this possible, and encouraging them to think big and seeing them accomplish great things is the best part of my job by far. So all the credit really goes to them, and hopefully I sort of went along and acknowledged them. And of course, big thank to all our collaborators and funding agencies. And that's right, we're hiring. So if you're looking for a postdoc, or you know of anyone that's looking for a postdoc, please have them reach out to me. And of course, the U24 for giving me a chance to share our work today, and all of you for attending. Thank you.
Thank you so much, Vic. That was wonderful. Taking over, Greg had to step away. So just want to thank you again and open the floor to any questions that anyone has.
Oh man, we made it on time. We have 5 minutes. That's great.
Oh, yeah. A little over 5 minutes. If anyone has a question, you can just unmute yourself. Ellen.
Oh, Ellen has a question.
Hey everybody, let's see if I can get my video on. Vicki, that was amazing, so beautiful, and really exciting work that I haven't seen yet. So that's really, really fun. So my question for you is regarding the oxytocin story, which is astounding, and I didn't realize that these hypothalamic neurons sent direct projections back to the cord. But I'm wondering, since oxytocin is also in the bloodstream, obviously, have you thought about how to sort of tease apart the contributions of the direct projections versus circulating oxytocin?
Yeah, it's such a important question, right? So as Ellen mentioned, there's these direct projections to the spinal cord. And there's a subset of the OT neurons that have direct projections that don't project anywhere else, which is interesting. So we're thinking of ways how to manipulate those specifically. But not a lot. Some work has been done in sort of, but there's some OT neurons that project the spinal cord and project anywhere else in the brain. And that we think is important, right? Because you have to coordinate multiple regions of the nervous system to get a desired effect. But as Ellen mentioned, these OT neurons also project to the pituitary, and there they release it through the bloodstream. So what we can do is manipulate the OT neurons that project directly to the spinal cord, and this is where we're starting as a stepping stone. Sometimes it can be really hard to disambiguate the effects of the bloodstream versus the descending. We're attempting to do that in some ways, but I think it's very hard. I think in the beginning, just identifying the OT neurons that project down to the spinal cord and seeing what time scales does it operate to alleviate pain. The way that I kind of like to think about it is that, through the bloodstream, you have sort of these basal levels of sort of oxytocin that your spinal cord sort of exists at. And then the descending oxytocin via direct projections can sort of like tip the balance one way or another. That's why it's sort of like a really complex system where you have excitation, you have inhibition, and it's just a matter of that collective ensemble of those output neurons. Hence why we turned more to like calcium imaging of those projection neurons to see how oxytocin modulates them. But yeah, I think it can be hard to disentangle both. But right now we're really keyed on to like, what are the descending projections? What is it that they're doing? But that's how we begin to think about it. Yeah.
Thank you.
Any other questions or comments? Okay, thank you.
So Vic, I have one, if you permit me a minute just to ask.
Yeah, absolutely.
Yeah, I thought that was a great talk, and really excited about the the complex behavior and the sort of naturalistic language processing you're leveraging to try to understand these complex pain-related behaviors. With the increase in accuracy to like 0.9 or 90% accuracy, do Do you hypothesize that there's going to be a ceiling around that level, just given that there's individual variation in animals that are undergoing whatever the procedure is that you're doing to put them into a pain state? Like, are we expecting this to ever be 100% accurate? Because I would argue that, as animal coders, we sometimes get things wrong because maybe some animals don't show the same level of behavior, after the manipulation. So I'm just curious what your thoughts are on that. Like, is there a ceiling that we're anticipating hitting before, 100% accuracy?
Yeah, that's such an important question. This idea of, as you start to get more and more of a bird's eye view on how these modules are connected with transition probabilities and different combinations, are you ever going to hit like 100%, right? And so I have two thoughts about that. So the first thought was, well, we know mathematically speaking, if you all of a sudden, take modules and transitions, it's going to be a priori much more accurate, right? So that was our hypothesis going in. We didn't know whether that particular embedding technique, Doc2Vec, was going to be, good or not. Like, we didn't know, but we thought, like, if there was a way to put these modules and transition probabilities together, it's bound to be more accurate. And indeed it is. So that was good. The second point, which is, I think, important for subsequent studies, as we start to do more pain models and analgesic models, is saying, what is a meaningful change? Like, if we can use these embedding techniques to look at combinations of modules and transitions, which is a priority, a behavior, right? So much like, if you have a knee injury, you're going to compensate, you're going to walk differently, which would be different than, say, you have an arm injury in which all of a sudden your reaching is impaired, right? But what are signatures of that pain condition? What are signatures of overall, being in pain? So, like, I think if you're preclinical, mouse, pain neurobiologist, you like to get at what are the common signatures of pain across all these different pain models, and what are unique signatures of pain to these different pain models and analgesic models. But I think, like with any of these technologies, they only get better the more models you have. And so in this paper that we're hopefully publishing soon, we have 2 models, but obviously in the future, it'd be really interesting to explore more and more pain models. And theoretically, there might be an embedding technique that would be eventually good enough for us to do our work and get good data to say, does this actually alleviate pain or did it not? And how does this look like in a nonverbal animal? Yeah.
Thanks. Look forward to reading that paper when it comes out.
Thank you.
Do we have any final question before we sign off?
I would ask one more, which is, how easy is it to adopt these 3-dimensional imaging, the depth coding imaging technique, and the computational tools?
Yeah. So from the standpoint of like the technical, sort of like the equipment is actually really easy because it's like a bucket and a Kinect camera. But now in terms of like, you get a bunch of data. So it's kind of like, when you have all your single cell sequencing data, it's all in like how you do the analysis. So Bob Data has I think it's a U19 or U20, it might be like basically to further develop this. And he has further developed it with like, different MoSeq reports, and everything's available on their website. I think the key is to have labs adapt it and use their own blend of embedding techniques to really understand. So one way in which we're starting to build it to take it to like the masses from our perspective, which is from a spinal cord perspective, is we're using it to scale, locomotor recovery after spinal cord injury, because there, like, the differences are so stark between uninjured and injured animals that you can really understand like which embedding techniques is going to really differentiate these different states. and then have sort of a proof of principle to say this is the best, or that is, so I think it's just a matter of more and more labs adapting this and collaborating. With that said, without a collaboration, it can be really hard, because I have a couple of computer scientists, but you definitely need more, right? And for a lab to have more than one or two, or even to have one, can be really hard. And so I think this is where sort of collaborating with Ishmael's group and collaborating with other groups has been instrumental for us to, to get this going. So I think this is going to take collaborative efforts via the NIH to really scale. And they put a lot of money into motion sequencing with Bob Data, and he has been able to get this to the masses pretty well. And also you can call them up. I mean, we sit with their developers all the time to ask them questions, and they're super amazing in terms of getting on Zoom and getting questions out, going through like paper revisions with us to address things appropriately, So I think it's it's there for people to use. Yeah.
Great, thank you.
Mm-hmm.
All right, I'm going to turn off the recording. Thank you all for being here, and thank you again, Vic, for such a cool talk.